Hello and welcome to the EASD 2020 first virtual electronic symposium entitled MiniMed 780G System Improving Clinical Outcomes While Reducing Diabetes Management Burden. My name is Ohad Cohen. I'm a professor of endocrinology and internal medicine, but also serve as the Director of Medical Affairs for Medtronic Diabetes in Europe, Middle East, and Africa, and it will be a pleasure to moderate this meeting, this symposium. Now, we will start with a short description of the need for automation in the treatment of type 1 diabetes, providing brand new data on the first year and a half of the first hybrid closed loop use in Europe and South Africa. It will be followed by a presentation presented by our Vice President for Product Innovation, Mr. Ali Dayanati. Now, he will tell us how from the millions of data points, hundred thousands of downloads and simulations, the new algorithm and the new product design of the 780G, the MiniMed 780G system was developed. Then we'll have the pleasure to hear Professor Tade Patelino. Professor Tade Patelino is if you don't know, he's a professor of pediatrics from the University of Ljubljana in Slovenia. And he also chairs the Department of Pediatric and Adolescent Endocrinology at the University Medical Center, again in Ljubljana, Slovenia. And he will discuss the first outcomes of the studies with this new MiniMed 780G system. And then we will end with a very interesting, I would say even personal insights from the experience Professor Amir Tirosh had with the use of the MiniMed 780G, and he will show us how this device actually changed not only the life of the people who are using this, but also their families, but most of all, even those HCPs who assist these patients to control the glycemia. Professor Amir Tirosh is a professor of medicine at the Tel Aviv University School of Medicine, and he heads the Institute of Endocrinology at the Sheba Medical Center in Israel. So, what is the need? Where comes the need for automating insulin delivery in type 1 diabetes? Well, it stems from the fact that we are used for failures. That means our patients, most of the patients treated with type 1 diabetes do not reach the glycemic goals that we want them to be. It's the well-known figure on the right from the type 1 exchange showing that less than 15% of patients with type 1 diabetes reach the goals that we want them to be from an A1C point of view. And that actually in the last 10 years, there's not been a very much improvement in goals obtaining these goals. And this is even though there's an increase of use of more technology like CGM use over time and new insulins and new analog insulins. But it's not only in the US. We can see that also in the UK, just published a couple of weeks ago from the NHS during the COVID epidemic, that less than 15% of patients in the UK are achieving the goals. So it's an issue which is global. And the question is, when we try to figure out what is the root cause for this failure, it's quite apparent that treating type 1 diabetes is very difficult. First of all, in order to get into good control, patients need to do something that I think no other disease requires them to do: count carbs. Now, counting carbs happens to be a very difficult topic to do. And 50% in one of the surveys of the patients say that it was one of the most burdensome aspects of treating type 1 diabetes. So it was not a finger pricking. It's not insulin injection. It's counting carbs, which is difficult. And you know what? Even if they do carb counts, almost 69% get it wrong. And it's not only the patients. If you ask HCPs, physicians, nurses, we also get it wrong. So counting carbs is difficult. And then when they need to correct the blood glucose, if the blood glucose is high, we have to make them go through a mathematical quiz. How high the glucose is, what is your target, do the math, divided by the sensitivity index, look at the trends. All this is a quiz. And again, there were some studies showing that they get the numbers wrong. Now, let's say they do get the numbers right, they do carbs correct, they do count carbs correctly. And again, it's only 2 out of myriad of factors who influence the level of blood glucose daily. And the most important thing is that there's a huge day-to-day variability. And as you can see on the left, it's the same patient. Actually, it's a woman who was with pre-gestational diabetes. One day her CGM reading is like day 1, the other is day 2, and you can see that there's a huge variability that needs to be addressed differently in different days. So how do the patients do this? They need then to do a lot of actions to be accurate. What are these actions? The first is getting more and more data on their glycemia. This data may be from frequent monitoring by SMBG, and we know, and you can see on the left, that the more frequent SMBGs per day are associated with better A1C. And in this particular study, more than 10 finger pricks a day were associated with A1C, which is close to 7. Having now scans, intermittent scans, make it easier for the patients because they don't have to prick their fingers, but still the numbers of scans per day that are associated with goals of glycemia are huge. For having an estimated A1C of less than 7, the requirement is to be associated with more than 22 scans a day, and for time in range, more than 39 scans per day. Now, this is a huge burden. Now, what is the outcome of this input of glycemia? Is bolusing or taking carbs. So we all know that the more frequent bolusing, the better control is. And this is from a century-old DCCT data showing that multiple daily injection is better than twice injection of mixes that were the control or the conventional therapy at that study. So more frequent measurements, more bolusing improves glycemic control. So at the end of the day of the patient with the people of type 1 diabetes looks like this. And this is only the load or the burden they have to treat the diabetes, and still there's life going on there. And this is what's called by one of the physicians in the UK, they call it peak burden. This is the burden on the patients with type 1 diabetes to get to good control. Now, if this is the burden, we're not surprised that it can lead to a what you called burnout. And this was apparent by the EDIC study that followed the DCCT. So once after the DCCT ended, and the closed monitoring of the patient stopped, and they came and they went back to the daily routine, daily lives, the A1C went back and increased beyond the A1C of the target of 7 and below. So it's very difficult for the patients. And the answer is, can automation play a role in making this burden easier while obtaining the targets of glycemia? So here comes the idea of the hybrid closed loop automation in insulin therapy. And it actually has the same components of the sensor augmented pump. You have the sensor, you have the pump, you have the infusion sets, you have the data management systems and the glucometers. But the issue is that you have an algorithm that provides the insulin injection to be coupled to the glycemic levels that is measured. And the amount of data points that are measured are 288. But that's not only the data that the algorithm has. It has also information about the insulin given, about the statistics of how much insulin was given in the last days, weeks, and so on. And the output is not just 3 or 4 injections per day, it can be up to 288 injections a day. So does this concept of having many inputs, many outputs, an algorithm that combines them together provides the solution to the issue of the failure in achieving glycemic control that we have with patients with type 1 diabetes? So the 670G was the first automated insulin delivery that reached the clinical realm. And what it did was actually automate the basal rate. So no more predefined basal rate. Basal rate is now defined every 5 minutes according to the dynamics of the glycemia, aiming to a blood glucose of 120 or 6.7 millimoles. And this is done according to the glycemia dynamics at that moment, but also taking into account the specific parameters of each patient. And this is adapted every midnight, every 24 hours. So the system is individualizing and personalizing the basal rate according to the patient's parameters and the patient's needs. So this is in theory. How does it look in the real world when it reaches clinical use? For this, a group of scientists and analysts had looked at the data that has been provided by patients who consented and uploaded the data on their regular routine work during the beginning of the use of the MiniMed 670G system in the last year, almost 2 years, from October 2019 to July 2020. The countries that data was able to be aggregated are seen in the flags in the right from Europe and South Africa. And we looked at the data into 2 periods. Period number 1 was the period that the patients used the system before enabling the auto mode. It's not their basal, it's not their background use. They might have used MDI, they might use different sensors or different pumps. This is when they're already on the 670G system, but before on manual mode, before putting on the auto mode. The second period was post auto mode enabling. That means the period that the patient had after he switched the auto mode on. So the only difference between the pre-auto mode and the post-auto mode is turning on the switch. So it's minimizing the effect of, let's say, more education, more insights into carb counting and so on. It's mainly difference is from turning on the algorithm. And another important part is that in order that the data will be valid for the glycemic control, at least 10 days where more than 10 days of data was required for each of these periods. All in all, we're looking at data from 15,151 users from 14 countries. What are the results? So if you look at the overall results, those who were using the system achieved a time in range of over 70%, 71.9%, with low time below range and low time above range. That means the average was beyond the target of time in range. And the GMI, it's the glucose management indicator, which is a proxy of the mean sensory blood glucose, and it's a proxy of the A1C. The mean GMI was 7%, as is the goal. So at least from an average point of view, using the device in a large population in real world achieved targets for the mean. But the question is, was this achievement done by countries that are, I don't want to say better patients, but maybe better food, better physicians, or was the success dependent on specifics of geographies, of knowledge? And here we look at what I call the Eurovision of the results of the 670G in Europe and South Africa. So you can see there's some differences, and some of the difference might even get to statistical significance. But what is really interesting is that no matter what language you spoke in that country, the algorithm worked. You can see that almost all the countries achieved either time in range above 70 or very close to 70. And on the GMI, almost all countries achieved levels of 11 and 7 or very close to 7, 7.1, for example. So it doesn't matter which language you speak to get to the outcomes you get from using automation. And if you want to go more deeply into what actually changed when you turned on the auto mode, let's just look at the pre-auto mode enabling as we discussed before, and if the same patients just after switching the switch on. And what we found here was, I think, is a very significant finding. So by just turning on the switch, the time in range increased by 10% from 62.5 to 72. But the point is that this is the first time we have a therapy for type 1 diabetes in which we increase time in range, decrease the GMI or A1C, but also decrease the time spent in the extremes. So you increase time in range, but decrease time below range and decrease time above range. Those kind of results we're not used to see in the past treating our patients with type 1 diabetes, because we either had a decrease in hypo by increasing the time in hyper, or decreasing the time in hyper by increasing the time in hypo. This is the first time we get statistical significant, but also clinical significance improvement in all the targets. Now, many countries in Europe have different, I would say, reimbursement schemes or decisions on who to put patients on certain devices. So we want to see if there was difference in different countries on the pre and post Autobot enabling results. And again, interesting, you can see that there's a different indications for different patients in different countries. But let's look at time in range. So even look at the different timing ranges before Autobot enabling different countries. Some were very high, some were very low. But at the end of the day, almost all reached target, irrespective of how you start the timing range. And the same with the GMI. You can see there were those who started with a relatively high A1C, or those who actually started with a low A1C, probably because of increase of hypoglycemia, all went to target of the GMI below 7. So what we're trying to see is that we can change the paradigm. We're used to failing, but this changes the picture. When you look at the obtainment of goals, either by GMI or by time in range, we can see that in this real world data, more than almost 60% of the patients, so it's the majority, more than 50% reach a GMI of lower than 70, and 61.4% reaches a time in range which is above 70. So from failing most of our patients, we go into a new era that we can actually succeed with most of our patients. Now, the question is it difficult? How long does it get to get this improvement? And I've already mentioned before, the only difference was between the pre-auto mode and post-auto mode is switching on the auto mode. And here we find a very interesting longitudinal data. So this is the same patients with at least 10 days of data from each period for almost a year. And what is very striking here is that the increase in time in range and the reduction in A1C is already apparent on day number 1. On month number 1. So in the first month, you already achieve the goals that are required. And this achievement is sustainable for the whole year. Again, first month, already getting to the goals, and this is already sustainable throughout the period of up to a year that we have the data. So in the next 2 slides that I want to give you some more information comes from a bit frustration on saying, so how come the algorithm succeeded where we, with all our years of experience, failed? So we try to answer this by looking at 2 groups, those with a GMI that was lower than 7%, before enabling Autobot, and those who were with a high A1C. And what we found out was that those with a lower A1C pre-Autobot, their time in range was very high to begin with because their A1C was well. But their time in hypoglycemia was almost reaching that goal that we don't want. That was close to 3.1%, close to the 4% that is the requirement. Turning on the auto mode did something incredible. It took the good, got them better. So even those groups increased the time in range from 75 to 78 by decreasing the time below range. Now look at those who had GMI above 8%. They were they were poorly controlled, and only 35.8% of time was in range before. And this increased by 20% to 58.8% time in range after just turning the auto mode on, reducing the A1C from 8.3 to close to 7, to 7.5. Now remember these numbers, you can say, okay, maybe we want more. We'll discuss this later when Ali will discuss the new algorithms. So this needs to be improved, but still 20% by turning on the auto mode, and almost no increase in hypoglycemia, but a major decrease in the hyperglycemia, which is the most dangerous for these patients, is incredible. But how did the algorithm do it? What was the secret of the algorithm that avoided us being so successful? And this is the last slide that I wanted to show you here, is that when we look at the therapy, that the insulin therapy that changed by turning on the auto mode in the two groups, these are the findings, which are, from my point of view, very interesting. First of all, both groups increased in total daily dose. And of course, those who started with a higher GMI, the increase of the total daily dose was higher, 24% versus 16.9%. So both groups added more insulin, but the partitioning between basal and bolus was different. Those with a GMI lower than 7, there was a decrease in the basal insulin percentage, while increase in bolus numbers. The opposite was in those with a high A1C. So those with a high A1C, the major improvement was increase in the basal insulin, which points out to the major cause for failure in the treatment of type 1 diabetes the way we do today, is the inability to give the precise amount of basal insulin, because every day is different. And by automating the basal rate, the algorithm was able to overcome this and to provide the insulin that is required at the time it was required. To summarize, we can say that looking at the data, we can get used to that most of patients using automated insulin delivery will succeed in getting to the GMI, the time in range, and the time below the range that we are aspiring to. They're using 81%, they're using the auto mode, so they are using automation. And the improvement is done very quickly after turning auto mode in the first month and is sustainable for a year. The system is successful because it adapts the basal insulin by individualizing and using the exact amount that the patient needs when it needs the insulin according to the algorithm. And with all this, it is a pleasure to me to hand over the presentation to Ali Dianati, who will discuss the new system, the MiniMed 780G. Ali, please.
Hello, and thank you for giving me the opportunity to speak to you today about the 780G system from Medtronic. My name is Ali Dianati, and I'm the Vice President of Product Innovation for Medtronic Diabetes. And today, our goal is to teach you a little bit about the need and for further advanced automation, specifically as it relates to the advanced hyperclosed loop algorithm. In addition to that, we'll cover the system connectivity and some of the new innovations that are coming. So just to get right into the details of what we're trying to achieve here with the 780G system, we take a day in the life of a person with diabetes. We know that it's filled with a series of decisions and work. Specifically, thinking about meals tends to be one of the more problematic areas for patients to recover from on a day-for-day basis. And a lot of the typical paradigms for handling bolusing, of course, doing it pre-meal, but then potentially under-bolusing for them so that you can correct later in case there are errors in that carbohydrate counting. Our aim with 780G, with the MiniMed 780G, is to eliminate the need for those corrections by bringing in more insulin and essentially correcting those higher ranges of glucose values within the patient's day-to-day activities and recovering from them accordingly. And so in a given day, a patient would go ahead and bolus for their meals, but not have to correct to recover from errors in carbohydrate counting, their differences in metabolism, or worse yet, undercounting their carbs. So if we take a second and look in a little bit more detail as to how the algorithm works, and we built this algorithm in conjunction with Dream Medical out of Israel, these autocorrections come into play every 5 minutes. And the intention here is to make sure that they help to prevent those highs. by effectively bringing in additional insulin beyond just basal insulin, of course, and allowing patients then to recover very nicely if they are going higher in the ranges of glycemia, of their glycemia. In addition to that, what patients want are to be able to stay in range without the risk of hypoglycemia. And this new algorithm allows for that as well. So think of it as being much more aggressive in its means of being able to provide insulin to patients without the risk of additional hypoglycemia. And so if we double click into what that looks like, and Professor Cohen went ahead and showed us some graphs on our existing system, the 670G system that does include that autobasal, we see when patients are recovering from a high glucose event, the system then tends to back off in terms of how much basal insulin it provides. And of course, when they're rising in terms of their glycemia, then the system provides more insulin, but it reaches a peak. And what I mean by that is in that 21 hour, if you will, we see that the autobasal delivery hits a maximum. And at that point, the system can no longer provide additional insulin without the user intervening or the patient intervening. So taking a step now with 780, one of the things that we have decided to do as a result of the advances in the algorithm is actually lower the target down to 100 milligrams per deciliter, or about 5.6 millimole. And in thinking about that a little bit, if we take what we had with the 670G, which had a target of 120, what this does is allow patients to stay in range more effectively because the algorithm is actually that much more precise. In addition to that, then, we bring in the notion of these autocorrections. And these purple bars that you see are when the system then provides essentially a smaller bolus, not the full need, but then accommodates and looks at their curves and how they are progressing with their glycemic variability, and adds in more insulin as needed. And the algorithm is smart enough to know that depending upon how they're trending, if they should increase that size of that bolus or decrease it, and effectively monitor it over a 5 minute to 5 minute to 5 minute period. And so as you can see at the 8 o'clock hour here, the patient was rising very quickly. And as a result of that, the system continuously provided additional bolus support in order to try to accommodate them back into range. So then taking a practical example for 1 second, here you see a 24-hour trend for a patient. And in the evening there, around 6 o'clock, they had a meal. And that meal they accommodated with about 50, assumed that it was about 50 grams of carbohydrates. But what we see is that later in the night, later in that evening, that they had under-bolused or under-accommodated the number of carbs. And therefore, the system went ahead and took over, and all these small purple lines you see on the bottom there, or boxes, I should say, are all the additional autocorrections that the system went ahead and put in. And as you can see over that 4.5 hour period, a number of these autocorrections came to play and came to bear for the patient, and more importantly, brought them right back into range in a tight window of time. So as you can see, the 780G system allows for a little bit more forgiving behavior in terms of being able to provide that insulin as patients need it. And of course, carb counting is not perfect, and therefore, with it, the system can make up for that. In addition to having this advanced algorithm, we're bringing in a number of improvements within the system itself as it relates to connectivity. So today, a patient with 780G will now have the means of having their own smartphone app and direct connecting that to the CareLink server, which then will allow a caregiver to have what we're calling CareLink Connect, which is the means of them being able to see what's happening with their diabetes management. In addition, the 780G has Bluetooth built into it and allows for that same uploading through the computer, but more importantly, also through the phone, not requiring the patients to have to go ahead and do that. So in simplistic terms, the smartphone connectivity allows the user to see what's been going on or what is happening within their day as it relates to their glycemic changes, how much insulin's on board, and of course, whether or not they are in SmartCard. A care partner would then get to see how that patient is progressing as well, inclusive of seeing their alarms and alerts. And for remote follow-up purposes, all of this data will automatically be uploaded into CareLink as a result of having that smartphone connectivity, so that when a remote follow-up is required, that data would be easily available and not require an additional step to upload it into CareLink. So in summary, the 780G system includes this basal insulin automation, just like it did in the 670G system, but now it also includes the corrections for bolus, the correction bolus automation as well. In addition to that, it comes with smartphone connectivity to allow patients, and a care partner app to allow care partners to see what's been happening with their day-to-day glycemic variability, what's happening with their insulin dosing, and whether or not they are in SmartCard. Because of this connectivity, automatic uploads to CareLink are now enabled. And last but not least, because of this Bluetooth connectivity as well, the pump is actually upgradable over the air, which means that future software upgrades will become a possibility with these pumps. And as new algorithms and sensors come into play, those upgrades will become available for patients. Lastly, we've made enhancements to CareLink to improve upon the working environment within it, so that healthcare practitioners have additional ease of use and more data available to themselves to interact and virtually speak and provide feedback to those patients. But then if we take a step further and look into the future a little bit, what we are also working on is extending the wear of the infusion set and the reservoir to improve the patient experience. So specifically, we're extending that wear to nearly twice as long, and in some days up to 7 days, to reduce that patient burden. So instead of having to change a set in about the infusion set in about a 3-day kind of time frame, we have now the capability of extending that up to 7 days. What this can allow for is the means for patients to also save on insulin, because of the fact that when we do these changeouts, insulin is wasted, and reducing them by a number of 2 or a factor of 2 effectively saves something in the order of 5 to 10 vials of insulin per year, and not to mention the patient burden that comes with that. In addition to that, we're working on improvements of the sensor as well, and so a new sensor algorithm will become available very, very shortly that allows for 0 calibrations and the means for patients to be able to dose directly off of their CGM. In addition to that, we've made some marked improvements in the way that its accuracy in the hypoglycemia range as well, which is, of course, the one area that patients desire the most to have the added accuracy. So I thank you for your time and hope that you're having a great EASD. Thank you.
One, please.
So thank you, Ali, for the simple explanation for us on the algorithm, but also thank you for simplifying the therapy for our patients. And I just want to remind you that there is you can provide your questions through the app for the Q&A sessions that will follow the end of the presentation. And now it's a great pleasure, Tade, to have you on board and provide us some of your inputs from the first clinical studies with the MiniMed 780G. Professor Patalino, please.
Hello, everybody, and sincere thanks for joining us today on this virtual symposium. I'm very sorry I cannot be with you live in person. I really miss live social contacts, as many of you do also, I believe. So let's do it in this virtual way and hope that we will meet very soon, also in person. Today, I would like to share with you our experience and experience for 2 other very recent studies on the very new MiniMed 780G system an advanced hybrid closed loop that also administers automatic correction boluses. The agenda for today is first the clinical overview of the United States pivotal study, followed by the New Zealand study, and finally by the NIH-funded FLAIR study. And we will discuss the clinical outcomes, both performance and safety, and also a little bit a glimpse of system usability and learnings that we had from this total very large clinical trial experience of the new advanced hybrid closed loop system that we just recently actually experienced. So the United States pivotal study with Anders Carlson as the principal investigator, safety and system optimal settings as a primary objective. It's a regulatory trial for the FDA. Age is 14 to 75 with an additional group from 7 to 13 years old that is still ongoing, followed by the New Zealand study with Martin de Bock as primary investigator, again, safety and performance, and the regulatory support for the CE mark, which in the end will learn the system already acquired. And here the population age range was 7 to 80. And finally, the NIH-funded FLAIR study with Richard Bergenstahl and Moshe Philippe, both from the United States and Israel, as co-primary investigators, with the primary objective of safety and performance in challenging populations. So typically a publicly funded trial, they don't want to exclude any group, as you will see. And the age range for 14 to 30, the most challenging part of the population with type 1 diabetes. And the advanced hybrid closed loop system used in this study was as a platform, the version 670, but with the algorithm 4.0, which means actually that the algorithm is the same as in the newest 780G system, but the hardware was the old one, so without the connectivity functionalities. And the version that we used is not planned to be commercialized, neither submitted for a CE mark. However, the new system, the MiniMed 780G, already received the CE mark, as I mentioned initially. So it is commercially available in the European Union. The pivotal study first. The design was a single arm, 16 centers in the United States, individuals with type 1 diabetes for equally or more than 2 years, aged more than 14 years old, with the additional group of 7 to 13 still ongoing, on therapy with a pump for more than 6 months with or without a continuous glucose monitoring, and A1C below 10. The primary endpoint was time in range. This is a very good news also, having time in range as a primary outcome, because it seems to be the most acceptable and also the most practical management tool in type 1 diabetes. And the advanced hybrid closed loop settings with a glucose target of 100 milligram per deciliter, 5.6 in millimolar, or 120, the old one if you want, and with the active insulin time as per physician's judgment. And as you can see, it had a run-in time of 14 days and then a crossover design. So first, the two groups used either 100 or 120 as the target for the closed loop system, and then they switched over vice versa. The participants, 157 in total, so a robust study, around 50% female, slightly more. The mean age, 38. Diabetes duration, well advanced on average with 22 years. BMI, very close to the normal range. Time in range before the trial, 69%, with an A1C of mean A1C of 7.5%. And this is the glycemic target. So I think a very impressive result if you want to focus your eyes first on the green, because this is what many people do. The green light actually an increase for roughly 69, as we said, to 74. So a really robust and per definition, per guideline definition, actually a clinically meaningful achievement that of course was also statistically significant. But with this, actually, importantly, the time below range, so the time that encompasses all ranges below 70 mg/cent, actually also significantly lower by 1%, which again is clinically meaningful and is statistically significant. And finally, the time above range, of course, also very significantly reduced by almost 5%. So again, very close to this clinically meaningful target, and of course, statistically significant. With this, also for those that still prefer to see the A1C, yes, 0.5% in this not very long period, so probably clinically a quite significant success, 0.5% less, with of course reaching clinical significance. If we compare adults in this study with adolescents, so adults is anything above 21 years, and adolescents 14 to 21, actually we see a surprisingly comparable results. So if you judge by the final outcome, adolescents in the green are almost as good as the adults. But if you look by the improvement, actually, the adolescents almost doubled it. So of quite, if you want, for some perhaps surprising success of the usually the most challenging group for actually any disease or even without the disease for adolescents, that actually achieved a result of a 10%, so double the target if you want, with this advanced hybrid closed loop system, again, with a very significant reduction in time above range and also a reduction in time below range. Same, of course, holds for the adult population. If you compare day and night, of course, the night was more successful because most people, at least in general, eat less during the night. And of course, this is also reflected in the success, a probably astonishing success of 81% of time in range during the night, but also the time in range during the day. Even if we take the more successful night away, it's still in target. It's still above 70, it's 72%, which of course is a significant result on its own, and just add actually to the whole significance of the trial. And this is the comparison between the target of 100 and target of 120. As you can see, the setting of a target of 100, which is 5.6 millimole per liter, was more successful. And this for some may be surprising, but what is actually surprising to probably most is that the time below range was the same. And this is quite impressive. So there was actually no statistical increase in hypoglycemia, yet the time in range was considerably better in the setting with 100, or if you want, 5.6 millimole per liter. So even if you use it into the more ambitious setting, into the more ambitious range of glucose, it is still robustly safe. Here, another way of presenting, so 47% of all participants achieved the glycosylated hemoglobin goal, which is, we all know, below 7. 84 of participants achieved the glucose management indicator goal. And this, of course, deserves special attention because in several recent publications, this metric actually may better describe mean glucose as compared to glycosylated hemoglobin. And finally, 79%, almost 80%, achieved the goal of time in range with the optimal setting of the target of 100 mg/cent of 5.6 millimole. If we now move to New Zealand, here the study actually had a slightly different design. It still was crossover, but it compared two different systems. It compared the advanced hybrid closed loop with the predictive low glucose suspend, a well-known system that is actually used all over the world by thousands of people with diabetes. Two centers in New Zealand, people with diabetes for more than a year, with an age range between 70 and 80, On therapy with a pump for more than 6 months, with or without CGM, very similar, and A1C same, below 10%. Primary endpoint, again, time in range, and the setting, again, either 120 or 100, with a fixed, in this case, insulin time of 3 hours. A run-in with a sensor-augmented pump, and then randomization to advanced hybrid closed loop or predicted low glucose suspend, and a switch, a crossover after 4 weeks with a 2 weeks washout period. Again, the population, a little bit more than 50% females, slightly lower age, 23, slightly lower diabetes duration, 13 years, but still considerable amount of years after the disease onset. BMI slightly lower, time in range, initial time in range lower, and slightly higher A1C. So a different clinical population. Let's see the outcomes. Again, this is the run-in, the control group, which was predicted low glucose suspend, the advanced hybrid closed loop, and additionally, the advanced hybrid closed loop with a target of 100. As you can see, also in this trial, the advanced hybrid closed loop in green, the time in range is within target. And the increase actually is more than 12%. So double what we describe as clinically meaningful. However, if you want to see separately the target of 100, again, even better than the total group. And the time below range here, if you want, again, improved, but not deteriorated with the use of 100 milligram per deciliter of 5.6 millimolar target. So again, the same message, even in the more ambitious setting, safe, robustly safe, and extremely successful. This is a glimpse, actually, on the patient-reported outcomes. As you can see, I did not put significance, but several of those are significant. As you can see, just as a glimpse, because this will be separately presented and published, you see that most people improved, actually, different aspects of the quality of life while using it. Finally, the FLAIR. Again, it's a publicly funded study by the National Institutes of Health, the NIDDK. It's a crossover design, randomized controlled, and actually here the comparator is the best commercially existing pump. It's the 670G by Medtronic. 7 centers in the United States, 2 in the European Union, and 1 in Israel. Individuals with type 1 diabetes for more than a year, aged 14 to 29, on pump therapy for more than 6 months, but also on MDI, and with an A1C from 7 to 11. So we really wanted a very, very broad inclusion for this population. The primary endpoint here is slightly different. It's time below 180 during the daytime and time below 54, which is, of course, 3.9 during the 24-hour period. So below 3 during the 24-hour period and below 10 in millimolar during the daytime. The target setting was 120 initially and reevaluated at the discretion of the physician. And the insulin action time, actually, it was 4 hours initially, and then it was decreased to 3 by the physician's judgment. As you can see, a much more conservative setting, also because of the population that was included. A run-in period, 3 months, either advanced hybrid closed loop or hybrid closed loop, 1 month washout, and the switch, the crossover. And here the group is, again, 113 people. Here, slightly more female, even 62%. A lower age, of course, because the group was on mean with a lower age at recruitment. Also lower duration of diabetes, but still, please see, more than 10 years on average. BMI within the normal, time in range initially lower than any of the 2 studies, and higher A1C compared to any of the previously demonstrated trials. Both primary endpoints were met, actually. Here, as you can see, run-in, the time in range during the day was 42, went down with hybrid closed loop to 37, and went additionally down with advanced hybrid closed loop to 33%. And this, of course, is likely due to the automated correction boluses that the advanced hybrid closed loop is delivering. And also the time below range, as you can see, very comparable. So exactly the same here in the run-in and in the advanced hybrid closed loop. So a very important, and I think a very welcome outcome, demonstrating robust safety of this system. Clinical outcomes, this is the time in range total. As you can see, actually, with the advanced hybrid closed loop, despite the fact that people on MDI and people with very, very difficult initial control were included, still almost to the target, 67%, with a mean increase of 4%. So again, in the clinically meaningful range in between the two groups. At the same time, time above range was diminished, and also, actually, there is no change, again, in time below range. And here you have, per age groups, as you will appreciate, actually, no difference in between the adolescent and the young adult group. Here, actually, the surprise is slightly less because most of us that actually have the privilege to meet these people with diabetes in this particular age group, we know that the challenges they face are considerable. And this success, of course, for this age group, we actually see as a very, very important step forward. If you want to see, again, the differences in the populations to better understand the outcomes that I just presented, the US pivotal, again, so we have a pediatric age group, so a pre-pubertal, if you want, age group that is still ongoing. We had an adolescent age group and an adult age group. Compared to the New Zealand trial, very similar. Here, the whole trial actually is already finished. However, compared to the FLAIR trial, it is a younger population. So we do actually the population 7 to 30. So what is usually seen as the population that needs most of our attention where we treat diabetes. Previous therapy, always an insulin pump in the US pivotal in New Zealand, whereas any previous therapy, including MDI, in the NIH-funded FLAIR trial. And the A1C, close to the target with the US pivotal, slightly above in New Zealand, and considerably above in the publicly funded trial, when we really wanted to be as broad at the inclusion as possible and as considered safe at that time. The time in range actually as well, 69, so very close to the target, actually even initially in the US pivotal, 59, so 10% less in the New Zealand trial and even lower in the FLAIR. And probably for the last part of my talk, the most important message, the severe hypoglycemia and DKA. In the US pivotal, no severe hypoglycemia, no DKA. In the New Zealand, no severe hypoglycemia reported and one episode of mild DKA. In FLAIR, one severe hypoglycemia reported, and it was a sad event, so it was not directly related to the new technology, and no DKA. So once again, despite the very ambitious setting, the most ambitious that the system actually offers, it was robustly safe. And here actually is the achievement on glucose management indicator. As I mentioned previously, this indicator may reflect the real mean better than glycosylated hemoglobin for several reasons. A beautiful paper from the Jocelyn group recently published in Diabetes Care. As you can see here, actually, even the FLAIR trial not only reached the target, but exceeded it, which I think is also a very interesting view on this data from a different angle. And this is for the time in range, so very, very good result in the US pivotal, high above the target, reaching the target slightly above in New Zealand, and almost reaching the target despite the broad inclusion criteria with all therapies, if you want, therapy modalities before the beginning for the FLAIR study. Time below range, all within target, FLAIR New Zealand on the half, if you want, on the target. So robustly safe with really minimal time below range. And this is actually time in auto mode, probably the most obvious result, if you want. So very, very high time in auto mode. US pivotal, 97%. New Zealand, 96%. Even the FLAIR with the population that included 86% in auto mode, which means that 86% of the time the system was functioning, was working, was helping people with diabetes manage their chronic condition. I would like to propose you a couple of take-home messages. The advanced hybrid closed-loop system is demonstrated to be robustly safe. Out of total 329 patients included into this randomized controlled trials, which is above 70 patient years, one severe hypoglycemia not related to the system but to a tragic event, one mild DKA related to site occlusion and a concomitant viral illness. It provided significant clinical improvement as per our definition in both glycosylated hemoglobin, time in range, in also glycemic management indicator, which we discussed separately. These improvements were observed in a broad population, even in the most challenging situations like adolescents with a mean A1C of 7.9 up to 11%, and previously on any therapy, including MDI. All participants spent a very high percentage of time in auto mode. The lowest was 86%, so really high percent of auto mode, so the time when the system was functioning without interruptions. And it seems that the best clinical outcomes are achieved when the advanced hybrid closed loop is at optimal setting, which is a target glucose of 100 mg/cent, which is 5.6 millimole, and active insulin time of 2 or maybe 3 hours, but it seems that 2 hours seems to be as safe and even slightly more successful. With this, I would like to really thank you very much for your attention, and I'm looking forward to discussions today online or later on at any time whenever we are able to meet again in person. Thank you.